Good morning, my name is Blanca Nega and I am a member of COVEMI Research Group of the Department of Animal Health in Complutense University of Madrid. I am going to present one of the works I am carrying out for my thesis called Detection of Antibiotic-Resistant Isolate of Clostridium perfringens in a free-range pig abattoir and processing plant. Microbiological safety in the food production chain from its origin on the farm or in the field until it reaches the consumer is one of public health key issues and it receives serious social concern. This is evidenced by the high level of media coverage of outbreaks of foodborne infections. There are many microorganisms involved in the transmission of disease through food. Among them all, today I am going to focus on a bacterium called Clostridium perfringens. This bacterial species is gram-positive, strictly anaerobic, and spore-forming. It is commonly found in the intestinal tract of humans and animals, but it can also be found widely distributed in the environment. It has been characterized as a pathogen responsible for causing some severe gastrointestinal disease in humans and animals, usually through the production of different toxins. These toxins allow us to classify the species into several toxinotypes, called by letters A to G. Studies on different foods of animal origin have shown the presence of Clostridia in these ones, suggesting that there is a potential risk that this bacteria could cause foodborne infections. Furthermore, there are some studies describing that multidrug-resistant strains of Clostridium perfringens have been isolated from food, which is an additional cause for concern in the current context of a generalized increase of antibiotic resistance in several pathogens. So, taking into account all the above-mentioned information, the objectives of the present study were to determine the antibiotic susceptibility of Clostridium perfringens isolate obtained from a free-range pig slaughterhouse and processing plant, and to identify those isolates considered multidrug resistant. In other words, those isolates that are resistant to at least 3 different classes of antibiotic agents. For this study, 85 isolates of Clostridium perfringens from a previous study of our research group were used. These isolates were obtained from samples taken at different steps of the processing chain of an Iberian pig slaughterhouse in Córdoba, Spain. To summarize, we will differentiate the origin of the isolate as animal, like carcass or intestinal contents, or environmental, like drugs or knives. Regarding the characteristic of this isolate from the previous study, 94% of the Clostridium perfringens isolate belong to toxinotype A, they produce alpha toxin, and 6% belong to toxinotype C, they produce alpha and beta toxin. 95% of the isolate of this species were from animal samples, and 5% were environmental. The antibiotic susceptibility of Clostridium perfringens isolate was analyzed using the EATS method. This is a commercial agar diffusion method that is widely used for strict anaerobic microorganisms, since it is simpler to implement than the reference methods proposed by international organizations. The method consists of the following steps. First, the isolate stored at minus 80 degrees were defrosted at room temperature and seated on blood agar Columbia plates. These plates were incubed at 37 degrees in anaerobic conditions for 3 days. After this time, those strains that had grown were reseated onto another Columbia blood agar plate to achieve optimal colony growth. These plates were again incubed at 37 degrees in anaerobic conditions for 2 days. In the next step, suspension of the isolate under study were preparing the brain hair broth, reaching in all cases a concentration equivalent to a McFarland 1 standard. These suspensions were sewed with a cotton swab on the surface of previously pre-reduced Brucella agar plates. Once the surface of the inoculate plate was dry, the EATS plastic strip containing a concentration gradient of a specific antibiotics were placed on them. All plates were incubed at 37 degrees in anaerobic conditions for 2 days. After this time, the results were read. In this technique, the value of the minimum inhibitory concentration must be read at the height where the halo of inhibition of bacterial growth cuts the strip. The results were collected in a database created in Microsoft Office for further analysis. The results of the study are discussed below. The Clostridium perfringens isolate were susceptible to amoxicillin clavulanate acid, rifampicin, and vancomycin, and they were resistant to the antibiotics shown in the graph. We can see a high percentage of resistance to imipenem, almost 50%, and resistance rates above 20% for penicillin G and tetracycline. For the rest of the antibiotics, resistance rates were below 20%. The most relevant resistance rates in our study were compared with previous studies by other authors. The resistance rates to penicillin G, tetracycline, and imipenem in our study were within the range of the resistance rates of most studies performed on pig and food isolates. When we searched for previous studies by other authors, also performed on Clostridium perfringens isolates from slaughterhouses, we found that there were hardly any in the literature. We could only compare with a study performed in a chicken slaughterhouse, where the percentage of resistance to penicillin G was lower and the resistance to tetracycline was higher than in our study. Regarding the multi-resistant results, we found that 30.6% of the Clostridium perfringens isolates were multidrug resistant. 92% of the multidrug resistant Clostridium perfringens isolate belonged to toxinotype A, and 80% belonged to toxinotype C. In terms of origin, 96% came from samples of animal origin, and 4% came from samples of environmental origin. The multidrug-resistant percentage obtained were compared with previous studies by other authors where multidrug-resistant isolates of different origin has also been identified. We did not find data about Clostridium perfringens multidrug-resistant isolate from slaughterhouses in the literature. The percentage of Clostridium perfringens multidrug-resistant isolate in our work was very similar to the percentage obtained in other studies carried out in pigs. Comparison of our multi-resistant percentage with those observed in cattle, chickens, food, or humans was complicated by the wide range of percentage, probably because the strains had different origins and geographical location, and the lack of information in the literature. In fact, although the number of multidrug-resistant isolates has increased in recent years, there are few studies that inform us of the prevalence of multidrug-resistant isolates of Clostridium perfringens in many countries and regions of the world. So, the conclusion of the present study were: further antibiotic susceptibility studies in slaughterhouses are necessary to achieve better control of multi-resistant Clostridium perfringens along the food chain and to prevent their zoonotic transmission. And, in particular, although very low or no resistance rates to metronidazole and vancomycin were detected in our study, given the great importance of these antibiotics in the treatment of Clostridium perfringens infection in human and veterinary medicine, we recommend to continue monitoring the susceptibility to these antibiotics. This is the bibliography used to carry out the study. Thank you so much for your attention.